Okay, so we're live now with our first book club of the summer for our Micro JC. And I'm going to turn it directly over to Karen, who is leading our discussion for the day. So Karen, over to you.
Hi, my name is Karen, and I'm a postdoc currently at Trinity Western University. And I'm also teaching a class in molecular genetics, but that's a little bit about me. But welcome to our microbiology journal club. Hi, Dave. Thanks for joining us. And so this is our summer book club. And in the summer, normally during the fall, we read different papers, select collection of different papers. But in the summer, we get this nice period of time where we can kind of indulge into science literature, and we start reading different types of books on our different topics. So one summer we had microbiology book, we had micro book club. Another summer but in the summer we read different types of books that kind of give us a perspective on what our science is like. And so that's mainly what I really enjoy about these microbiology book clubs, is that we start to in books, they are more able to delve into the history and how things have developed over time. And these are things that normally we don't get to read about. So it's exciting for me to get into these books. But now I'm going to be facilitating today, but without further ado, what I will do is let everyone introduce themselves. And so the person right next to me is Sangeeta. Is that how you pronounce your name? So I'll let Sangeeta introduce herself.
It's close enough. Sangeeta, my name is Sangeeta.
Oh, and tell us a little bit about where you are.
Sure.
In the book.
Yeah, so I'm a postdoc at Imperial and I'm working in epidemiology. I have just finished my PhD, and my PhD was in phylogenetics. So I am very much interested in genetics, etc. And when I heard about this book club, I love reading. So it was like, okay, I should be here. And that's why I'm here.
Well, thank you for joining us. We really appreciate having somebody. We're always welcome to have somebody new join our book club.
Thank you.
Okay, so next to Sangeeta is Nicole. Nicole.
Hey everyone, I'm Nicole Suktio, and I'm a postdoc at University of Northern British Columbia. So it's interesting to know that Karen is now 200 kilometers closer to me than you were originally before you defended your PhD. That's cool. I work on forest soil microbial ecology using amplicon sequencing methods, and I will also be teaching microbiology in 2018, apparently. So reading about genes is a bit of a departure from that, but at the same time, this book that we are going to discuss does sort of dive into some of the bacterial systems that allowed us to sort of pick apart what DNA was doing and the mechanisms around how genes are expressed. So I'm sure we will get into that eventually.
All right, thanks, Nicole. And next to Nicole is Laura.
Had to unmute myself. Okay, I'm Laura Williams. I'm an assistant professor at Providence College in Providence, Rhode Island. We're in the midst of our summer research program, and right now what we're focused on is our research with predatory bacteria. We also have done some microbiome projects. Those students have graduated, so now it's up to me to get those papers out, which is another thing I'm working on. But then I did want to say, because we've got two people who it sounds like just defended their PhDs, both Sangeeta and Karen. So yay, congratulations. That's a big accomplishment. So we should both be very happy about that.
Okay, and thanks, Laura. I already mentioned this. I don't feel it yet, but that's okay. I'm sure it'll sink in over time. So next to Laura is Kat.
Hi, I'm Kat Milligan-Myrie. I'm at the University of Alaska Anchorage, and I'm an assistant professor. I study how the host genetic background contributes to microbial communities and how the immune system interacts with those microbes. I'm a microbiologist at heart, and that's what I was trained at, and I've become an evolutionary biologist, kicking and screaming. So this was a good introduction or reintroduction or solidification of that genetic material that I should be an expert in and am slowly becoming at least knowledgeable in. So this was a great book for reading at this point in time.
All right, thanks, Kat. And lastly, we have Dave.
Hey, Dave Baltris. I just got the kids out into the car, so that was that ruckus. And I'm also back from like a red-eye flight last night, so I was finishing reading it on the flight. I'm a professor in Arizona. Started kind of reverse of Kat, so I was an evolutionary biologist, and then less than regretful, I kind of went into microbiology because it was easier than other things in terms of being in the lab and whatnot. So not kicking and screaming, but I became a microbiologist in grad school, basically. I am all about sci history books and really like the Emperor of All Maladies. So the Mukherjee book was kind of I was looking forward to this one. So, but yeah, so that's my background.
All right, thanks, Dave. I hope so now that we've gotten to know everybody a bit, I hope we'll have a great discussion. And thank you all for coming. This is one of the times where we have lots of people in the group. And so I think we'll have a great discussion here. And so one of the things that I do want to point out and talk about as a facilitator of this discussion, which is that this book covers a lot of controversial topics that can invoke very strong emotions in people. And so I just want to be sensitive to that. And I want to make sure that we can all have a rigorous academic discussion, but yet also be kind and respectful of everyone's opinions. And so that's just one of the things that I wanted to say before we get started. And so the book, without further ado, let's introduce the book that we actually read, which is called The Gene. It's actually if you look at it, it's a very thick book, but it's by Siddhartha Mukherjee. And he also, as Dave mentioned, he also wrote The Emperor of All Maladies, which is a book on cancer. So you can also kind of see those sort of things flowing through this book. And what I'll do right now is I'll just give a brief summary of the book and divide it into sections. But overall, The Gene, it talks about mainly genetics. So the history of genetics and how that biology of who we are also is inseparable from our social identities. And so he delves into multiple topics regarding just the history of discovery, but then the impact that it's created throughout society because of the ideas that because of the genetic ideas that have been uncovered in terms of discovery. And so if I were to divide the book, what I'll do for this particular session is divide the book into different sections and then kind of give everyone an opportunity to say, okay, what was poignant for them about particular sections and to go through it that way. We might not finish everything. And I realize that because it was such a long book, not everybody finished the whole book, and that's totally okay. If you do have anything that you want to add or say, I think that's part of the exciting things about having book club is we get all these interactions and different opinions and discussions. And so if I were to divide the book into sections, the first part I would say is probably about the history and discovery of the central dogma. The second section is more about the applications and knowledge that had been distilled from the first section. So there was sections about the creation of Genentech and different patent applications for insulin. And the third section was more about genome mapping and how the Human Genome Project began. The fourth section was definitely the most challenging section, which is the intersection of genetics, cancer, mental illness, sexual identity, and orientation. And the last section is mainly about what I would call cautionary and exciting tales of gene therapy. And so I'll start with the first section, which is the history and discovery of the central dogma. And so what the author goes through very elegantly is these sections discussing the story of Darwin, the story of Mendel. And so I'll just open up the floor for anything that people want to discuss in terms of that particular history section or things that you found interesting or stood out to you.
So I'll start. I'm actually going to maybe render an unpopular opinion. I actually think this book was two books. I think there was a book here that was all about the history of genetics, and I thought it was great. And I thought it was really nice for me to have a chance to sit down and read that and refresh my memory. But I felt like that was one book. And then there was a second book that was about kind of the latter things that you were talking about, Karen, like how gene therapy is now being developed. What are we learning about? There's a couple things that we might get into where he discusses, I think in a useful way, you know, penetrance, the idea of you could have this allele, but that does not guarantee that you're going to get this disease. And how do we understand gene-environment interactions? And so I almost feel like you could almost chop the book in half and make 2 books out of it. And it all comes down to, from an author's perspective, what is it that he wanted to do? Was it, I don't know that it was absolutely essential to walk through the history of genetics to get to discussing gene therapy and gene-environment interactions. I liked reading that part, but I don't know. I felt like these were 2 separate books. That is my point.
I think it comes down to so the first book or so, and I agree, but the first book or so is basically stuff that's been hashed over a bunch of times in a lot of good ways. But I don't know. My impression was that that's the book he wanted to write, and then he wanted to get into the other stuff that was more meandering a little bit. Maybe, you know, because you kind of have to talk about that stuff, but it's less hashed out, right? And so there's less, the stories aren't as well known, and there's not as much detail to it. So it was kind of a compromise between what things that would be a good book, but an ground that had been well tread already, versus like the new stuff that you kind of had to, you needed the background to actually get into. But I think the interesting thing to me, though, and just so having taught this, not this, but having taught kind of genetics for a while, the word gene actually ends up being a very interesting thing, because there's no good definition for it anymore. There was a great definition back before we knew about genetics, but now there's no good definition for gene that kind of covers everything, because it's all versed in phenotypes, and then once you start actually understanding like what the underlying genes and regulation and terminators and whatnot, all of those things together, you can have complete discussions about like where a gene stops and where a gene ends, because it's not, you know, it's one of those, like, it makes less sense the more you know about it a little bit. So it's kind of an interesting, just historical side note there. But
so maybe it should have been, the title should have been like The Gene ? The Gene? I don't know.
Just sorry for real quick. I just, in terms of from a perspective of non-geneticist, and when you talk about incorporating this encompassing definition of a gene, how has that changed in terms of ambiguities? Like, are you talking about like incorporating the are the regulatory?
Well, so gene ends up being, so when you don't understand when you don't know what the underlying basis is of phenotype. So the genes was coined, and he goes over this in the beginning of the book, but gene was coined when, in a strictly phenotypic world, where the point was to have something analogous to an atom, like some non-breakupable unit of inheritance. And so that works when you don't have to think about what the unit of inheritance is. You can just kind of theorize that it exists. But now that you know that there's proteins and RNA and DNA and RNA and the central dogma, because gene is rooted in phenotype, and phenotype, there's tons of underlying genetics that could lead to the same phenotype, it ends up being, there's just no good definition of the word gene anymore, aside from that one rooted strictly in phenotype. So when I teach it, I basically teach it as a gene is not start codon to stop codon. It's not necessarily promoter to terminator. It's whatever piece of DNA or RNA you need to complement a phenotype, right? And so that changes in different contexts. But I think we're taught all the way basically up through like intro biology, even in college now, that gene is, effectively start codon to stop codon, and they make proteins, and you kind of have to do that to simplify it. But when you start getting into the details, it ends up being this philosophical discussion about like even the simple word of gene is this complicated, arguable point, which is kind of it's
I like the fact that the book is called that, because it does kind of open up the, you know, we know a lot, but we don't really know a lot kind of aspects to it. But
all right, thanks, Dave. I didn't realize that, so it was good that you explained that. Sangeeta, did you have something to add?
I was just going to reiterate what everyone else was saying, was that I did not enjoy the first part of the book, and especially as David pointed out, other people have written about it. And I thought it's probably because I have read that and I'm familiar with that part of the story. That's probably why I was getting bored. But I guess I guess everyone else is also getting the same feeling. But I did enjoy the second part. In fact, I missed a couple of stations on my way back because I was kind of lost in the story. So yeah, I did enjoy. And I, in fact, I wanted to read this book because I wanted to read about those discussions about how genes determine our identity and how much of a role they play, the discussion about nature versus nurture. So for me, that part of the book is more valuable, more enjoyable.
Kat, did you want to?
Yeah, I actually have not read a lot of the history of the the background of the genetic information. And so I was really excited to read the first half, and I absorbed it in a couple of plane rides. Just came back from a trip. And so I read the first two parts. I just inhaled it and thought it was really interesting. But I had not read the in detail background on Mendel and all that stuff. I read, what you read in your basic textbook, but I hadn't really read deeper into that. So I found it really interesting, and I thought it put the rest of the book kind of in context. One thing I did want to say, though, is that I was really disappointed that there were no female geneticists before Franklin, apparently, and that he spent a lot of time going into this really great personal detail about the personalities of people early in genetics and their history and where they came from. And then that later on in the book is just kind of all that really rich history that puts these researchers in context is gone. And so you go from getting this really great history and in-depth analysis of where these people came from and how they influenced how they approached their studies, to this really brief, like, this is what they found, this is what they found, this is what they found. And I was a little bummed about that, and now I'm going to have to go and find some more in-depth books on some of the more recent female geneticists, because apparently we've only existed recently.
Well, I think the fact that Barbara McClintock is reduced to a footnote.
Yes! that was so frustrating to me. That was so frustrating.
You're right in that it becomes kind of a glossing over thing, but I think there's a problem of timing in the book where he does get into female geneticists before Franklin, but it's in the context of like the chromosomes, and you just don't get the appreciation in the build-up story, like you said, and it's there and not as in depth as that first half. Yeah, but no, there's a lot of missing points.
But I think, and that's why I started out by saying I feel like this is two books, and Kat pointed out the difference in style too, and just the way he wrote them. The two themes, I think, are even written and approached very differently. And so I don't know what the process was by which he wrote this, but I really think you could have taken this and made two books out of it. One of the things I did want to say that I did appreciate reading through the history, though, is it is very reassuring that these giants in the field really stunk at taking tests. It's like Mendel tried to take this exam and he failed it. And then he tried to take it again and he failed it again. He basically stunk at this. So you kind of feel reassured. You're like, okay, all right, that's good. I feel good about myself now.
Well, and the role that luck had to play with all this, like Wallace, who really basically did the same thing that that Darwin was working on, but it lost all of his samples, and there was a big catastrophe after a big catastrophe, and that's why you know Darwin's name and not Wallace's name. And I think that's a good way to introduce young scientists into the real scientific world. And this, some of it's hard work, and there's these ongoing discussions on Twitter about what's work and what's luck in research. And I think this is a great way to introduce the fact that there's a whole lot of luck in research.
Yeah, I agree with Kat too. Because I hadn't read all the history before, so I really enjoyed that. And part, I started, and what Laura was saying, like the fact that Mendel had failed his test. So I used it as one of, as I was reading the book, I used it as a way to encourage the undergrads that I have in my lab, because a lot of them are saying, oh, maybe my GPA is not good enough to get into grad school. And I'm just trying to tell them, look, grad school is all about, it's like running a marathon. It's about perseverance and it's about endurance. It's not necessarily about how well you can take an exam. And so I think that was helpful for them to see that, you know, even the father of genetics was had difficulty doing these things that they had difficulty doing as well. All right, Nicole, did you have? Oh, sorry, Sangeeta, did you have something to add?
Yeah, I was just going to add that even a more relevant example than Mendel would be that guy later on in the book who went on to win a Nobel Prize. I forget his name, who was 250th in the class of 250, and then he ended up winning a Nobel Prize. So that would be a good example to motivate students, I guess.
Yeah, I think that there's it's like Mukherjee was saying in the book, that the traits that this educational system selects for specific types of traits, and it's all about whether or not your traits or special skills adapt to a specific environment. And so if these people who know how to take exams can adapt to this specific environment, but that's not necessarily a determinant for whether they'll succeed in research. All right, so I'll move on to the next section that I was talking about in terms of the applications and knowledge or maybe I could probably lump those together. So applications of the knowledge in terms of the creation of Genentech, and an interesting topic which is fairly relevant right now in terms of patenting genes and in terms of patents and commercialization of genetic discoveries, and then also genome mapping and the Human Genome Project. So it's a lot of topics lumped together, but I just wanted to I'll just open up the floor and see what everyone has in terms to add for that.
Are you thinking about like which sections in particular are you? I'm casting my mind back to like a couple weeks ago.
Like so in terms of part 2, where they start to talk about some of it's the dreams of geneticists.
Okay.
That's what I was talking about. Like how Craig Venter created this whole other company just to compete or to work on the story of insulin, and how they ended up cloning that gene. Was it somasticin? I can't remember what it's called. But they cloned the smaller gene, and then they ended up attacking insulin, and there was that race, and then which generated the first possible patent in terms of genetics.
Mm-hmm. Got it. Yep. Yeah, I think there's a lot. I mean, there's so many ways you could have organized this, because one very obvious current. Eventually, the book gets into CRISPR, like one of the last chapters.
Yeah.
And what he talks about here in terms of the legal ramifications definitely is ongoing right now, because there's a battle for who gets the patents for CRISPR, who is the person who's advancing that discovery along, and what's the timeline. Yeah, you could get into this a lot about can you patent genes. So the motivation for the Human Genome Project funded by the government, and then the motivation for a company coming in to say, we're going to try to make money off of this. And I remember one of the things when I was reading this section was because the government is getting funding through taxes, and then the company, they kept talking about, or in the book he mentions the investors. And I remember thinking to myself, I'm going to have to read something about that, because who was investing in these companies and what did they think they were going to get out of them? Like, I know very little about the business side of, they're the ones pushing, I assume, to like, we should patent these genes or we should patent these technologies. And I'm kind of curious how that all ended up playing out. Like, what was the deliverable or the product that investors, just not from a science standpoint, but just from strictly an investment standpoint, what did they get out of it? I don't actually know.
Well, I think a lot of it. Oh, sorry. I think a lot of it is just being able to sell kits that help you determine whether you're going to have the diseases that he talks about in the epilogue and leading up to it. You know, there are all these diseases that are that have a very important genetic component to them. Different types of cancers, different types of I mean, he mentions them all in the book, but I thought part of the things that they were selling to investors is the ability to sell a kit that you would be the sole owner of the reagents needed to identify whether somebody had a genetic mutation. And given the ridiculous amount of money that we put into healthcare, this becomes really valuable to people who want to make money from being able to diagnose these tests. And then I thought that it was interesting that he brings in this whole idea of, do we really want that as a society? And I hope we get to that towards the end. But yeah, I think the initial thing, it's having worked in Madison for a while and watched these biotech companies emerge and then crash and then emerge and then crash. And a lot of what I've seen driving the investments is how profitable will this be in the end? Can we sell this particular primer set to identify this BCR1 gene mutation, to identify this mutation and whatnot? And I think that one of the things I like about this is that he brings in the fact that it's not just the genes, and going back to what Dave was talking about earlier, it's not just the gene, it's the gene by environment interaction that you have to consider. So you can tell somebody, well, yeah, you have this mutation, and so you're much more likely to have this particular disease or pass this disease on to your kid. But knowing that what role the environment plays, I think would change those investments as well. Maybe not.
Dave? Oh, sorry, Nicole.
Okay, so the Asylumar conference, I think, was the thing that kind of blew my mind a lot in this section. It devolved into a yelling garbage fire, apparently, which is not that surprising. And I mean, the details of it are a little lost on me, except for the whole late night drafting of a document to kind of come up with what are good conditions for moratorium or constraining use of this technology, which is really kind of something I think about now, because when you think about at least Genome Canada funded projects, there's always this policy component. And I don't think there were any policymakers in the room when Asylumar was going on, which I hope I'm correct about that. So, I mean, it's interesting to think about the burden of being a researcher immersed in this field, immersed in the details of this field, and now trying to think about societal implications. Because, I mean, number one, your head's in a very technical space, but also trying to think about, have I unpacked all the ethics with this? And the fact that it was actually students who were questions about, well, what is the potential harm, or how far can this escalate? So, and everything was actually happening really fast. I mean, to make insulin just on the heels of having the mechanics of doing recombinant DNA technology, and recombinant DNA technology that wasn't yet optimized. Like, we buy everything in kits now. So this was like iGEM on hard mode, to make drugs, to make proteins. It's actually kind of makes me stand back and wonder at some of the things that they went for that did work. For example, getting the clotting factor next of HIV becoming epidemic, and realizing that, at that point, I don't think they even had the antibody test worked out yet. I think we actually had good screening maybe by 1990. So there's a lot of things I wonder about. And I mean, the author doesn't really unpack a lot of the policy things, but I kind of wonder a lot about what people like Boyer and Cohen and their colleagues were discussing in terms of how far, when to use, when not to use, what are the range of diseases that are good targets. Because, I mean, the other thing that they knew that the public probably didn't know, even when the human genome was published, was appreciating the extent of how many diseases have multifactorial etiologies that might be cumulative impacts of the lifespan. So, I mean, we're picking things again, and Siddhartha Mukherjee reinforces this multiple times in the book about how our best targets for prenatal screening, our best targets for gene therapy, and our best targets for even identifying and predicting disease really depend on these strong penetrant monogenic pathologies, which were good, good resources to back to give initial credibility to recombinant DNA technology in medicine. So I'm going to leave it at that because I don't really know what else to say.
Well, I liked a lot. Oh, sorry, Dave, did you have something to say?
Yeah, I just want, it's like one line in the book, but it kind of hits home just being so there's part of the Silomar conferences, I think it was the second one. There's a line about the plasmid guys getting screwed. Like, where I can imagine, like, you've based your whole like research career on developing these cool new technologies, and then like everybody else just shuts you down with moratoriums and whatnot. It's I mean, they did okay in the end, but I can see how it was like it would be very frustrating if to have everybody else tell you what you can and can't do. And I mean, on the other side too, there's the precautionary principle always kind of plays into it, where who knows what, even with CRISPR stuff now, I mean, it's, you got to weigh the unknown versus, as scientists, and I speak for myself, but I can give my impressions of other people, when we have opinions and when we tend to think things, we think they're right versus, it takes some other people arguing with us to make us see the other side sometimes. So, I can imagine why these meetings were so heated, is because there was a bunch of like very loud, opinionated people in the room that all thought they were right, and it was determining the future, the next 40 years of what was going on. So, yeah, and the personalities were not necessarily the calmest of personalities too that were arguing over these things. So.
Yeah, no, I definitely agree that as scientists, that we have a certain ethical blindness from overexposure. And it's because, like, I remember that when as an undergrad, I was concerned about genetic cloning, but then as soon as I entered the lab and saw how easy it was, okay, I just put all these clear colorless solutions into this tube and it happens for me, I started to lose that perspective, and I don't have it anymore. And so so when Nicole was talking about, these students were the ones that brought up those interesting perspectives, and they were seeing far, far into the future compared to, when you're every day being in the lab. It's very hard for me to think about beyond these implications of what my research can go. And so Laura had mentioned earlier, that it would have been good to have someone who was a science enthusiast that read this book to help them give us perspective of what they thought. And the one more thing that I wanted to bring up was the fact that scientists were able to regulate themselves. I thought that was interesting, that they did, even though it was part of it, but they were able to stop themselves and think, okay, there is something that needs to be done, and come together and actually establish this policy and regulations. And I only.
But did they though? I mean, because they found loopholes, right? So it's, yes. You set up the regulations, but then you just do it with the private companies, and then it works out okay. So.
But it wasn't the same people that came up with the regulations that also did those loopholes. But I mean, in general, like those rules still stand as the rules that they came up with in terms of biological containment still stand as they do today. Is that correct?
Oh, frustratingly so. Yeah. I have to, anytime we walk a Arabidopsis out in the desert here between buildings, we have to cover it up and make it airtight, because what happens if a Arabidopsis gets out in Arizona and takes over the place? It's
oh wow, I didn't know that.
Yeah, it's I mean, but on the other side, I think there was I don't know if this is published or not, but if you go to Berkeley, I want to say, but probably any campus, you can go and isolate nematodes from the soil around like the lab buildings, and you'll always find like GFP nematodes in the soil. So
that's hilarious.
Yeah. So the regulations are there, but
it's glowing nematodes. I think Sangeeta had something to say too. I've seen her hand go up a couple times.
I was also going to reiterate the same point, because I was also very impressed with the fact that the scientific community came together to regulate themselves. And whether or not they did a good job at it, it was the intention itself, which I think that's the strength of science, and the fact that they invited not only scientists but also other community like writers, journalists, etc., to think things through. And I wish political leadership all over the world would do the same thing. Then probably we would have a better world. Before doing something, let's just think about what we are doing and where we will go with this. You know. So I really liked, and I completely agree with Karen that this was a very good thing that we have done. I think we should be proud of ourselves as scientists.
Yeah, I want to thank Nicole for bringing that up. So I guess the next section that I want to move into is more about genetics of cancer, mental illness, sexual identity, and orientation. And I think that was the meat of the confusion in terms of he asked a lot of questions and there weren't a lot of answers. And so I just, in terms of, like, he also talked about, he gave a lot of anecdotal stories and then also gave the corresponding academic stories in terms of how those two relate. And so I just want to, does anyone have anything in terms of, in particular, that they thought was really interesting in that aspect?
I'm always the first person to unmute myself. But I think I said this on Twitter, and I don't know how many people got to this part of the book, but like, especially into and this may be a different section that Karen wants to feature, but as I got into this latter part of the book, boy, I had thoughts. So here's some of the things that I really, and I think I might have already mentioned this, but some of the things that I think he did well was to continue to emphasize really heavily what people have already mentioned, to introduce the concept of penetrance, to introduce the concept of gene-environment interactions. So my hope is that if someone who's just interested in genetics but not a scientist picked this book up, by the end of it, they would go, okay, I understand now why this is very complicated. Why, if I go to 23andMe and get genomic data back, X does not equal Y. Like, I hope that that's something that comes across to people in this, because I do think that's a complicated topic that can be hard to understand, but I think he really hammers it here, which I think is good. Part of what I think he did a really good job trying to address, but this is where I actually think, okay, let me back up and explain. I think he did a very good job trying to bring into the discussions of this, the idea of there is a social construct for talking about things like identity. I mean, he's got one section where he talks about, you know, the genetics of normalcy, you know, and he spends a lot of time very carefully discussing normalcy as a concept that we as a society kind of decide upon, or that in some cases just ends up getting imposed on other people. You could very much talk to many people in the autism community who will have a lot of thoughts for you on what is a cure for autism. Do autistic people need to be cured? Do they just need to have more understanding about the way they interact with the world? And I think he does get into that here. I think this is a place in the book where he would have benefited from maybe being more like a reporter and consulting some experts, bringing in some perspectives from experts in the fields to kind of give a little more detail about some of those ideas. Because there are people who, I mean, this is their whole wheelhouse, is looking at how genetics and society intersect. And I'm kind of thought, I don't want to draw comparisons in this way, but I think this is the difference between, maybe this is the difference between a scientist writing a book and then somebody like Ed Yong, who is a science writer and a reporter who writes a book. And so I think this book was written from the perspective of a practicing scientist, but I think when you're getting into these really in-depth issues, I would have liked to have seen more featured about people who, this is their research, this is their wheelhouse, and they could kind of flesh out some of those perspectives. That was my thought on it.
So I'm going to go, and I'm going to say that I agree with you, Laura, on a lot of that, because something that really struck me towards, I guess, the last third of the book, including one of the author's ideas, which is articulated at least 2 points in the book, which I could not find, Mark, before I got here, is that Siddhartha Mukherjee seems to be wanting to articulate more about inclusive policy, but doesn't quite get there. So, I mean, so me saying that doesn't make a whole lot of sense. I'm going to unpack it with the discussion of the neuronal transmission gene HTLRRP. I did mark it in here, so I can get the page somewhere, I hope. Yeah, page 460, HTTLRP. So he talks about that study that had the control and the intervention in sort of the African-American population, where they had sort of risk mitigating sort of mentorship for this cohort of children. And I mean, it was a control and an intervention administered sample set where they didn't really know. I mean, it was a mixed population of people who had the short variant, which of course has sort of a highly variable phenotype that translates into higher propensity towards higher risk behaviors, trauma. And they were dealing with two populations that were equalized for socioeconomic situations and life situations due to, things that would have, of course, greatly enhanced, if this genetic propensity was there, sort of the worst of the worst outcomes. And it was interesting to see that when these educational options and guiding people to have sort of the coping toolkit or repertoire of what they should do in situations or how to take care of themselves, that those with the risk propensity genotype were more responsive to these interventions. So he's not quite saying it, but I mean, by inserting that example, he's very careful to make this point that if we are still at a stage, which we are at this point, of not quite figuring out the manipulations we need to mitigate risk and decrease suffering by direct manipulations of the genome, that we can always manipulate the environments that human societies provide to people. And that other thing that he mentioned at least twice in the book was that the deleteriousness of a phenotype really is very much defined by our interaction with the environment. And I mean, there's a lot of ontology in the disability rights sector, sort of. So I've read a little bit of this, but I'm not an expert, that does talk about disability as a social construct where the configuration of society and infrastructure and how easy it allows somebody with a disability or a different configuration of their range of abilities to actually function relative to somebody who doesn't have that same configuration are actually what keep place. So manipulating educational opportunities in terms of format of delivery, having caregiving for children with high risk, genotypes such as this HTTLRP, who might have had unstable family situations through going through the foster system, but having stable parenting, education, things that we can do if we can get people in the right situation to thrive in spite of what's biochemically there are feasible, because there are things that we can do that don't require manipulation of genes. So I think that he would have benefited definitely from employing the knowledge of people who, like you said, think about this intersection of genomics and society and genomics and policy a bit more, because there was a bud of that trying to come through in the last section of the book, and I don't think it was made as apparent as it could be. Although the other part of this, for a non-expert, that was very valuable, which is understanding that thinking about genetics translating into predicted phenotypes is very much highly probabilistic once we start to think about the environment-gene interaction and the fact that there might be polygenic genotypes predisposing, and also different temporal and developmental triggers as well as environmental triggers really prevents us from dealing with diseases that fall out of being easily explained by the central dogma, really. So that's what I have to say.
I think there's a couple of points that hit me in this part. One less controversial than the other, but the more controversial one. Every time, like the last few months, when somebody gets up on TV and says like,
you've got a Y chromosome, you're a guy. Like, I think this is one of the better, faults aside in how he dealt with some of the issues, but this is one of the better, like, 2 or 3 page places where you might be able to point people to say that things like gender and identity are not as concrete as, an intro bio textbook tells you. And I haven't seen a better discussion of that in other pop genetics books lately. Especially given the, SRY matters, but other things do too. And it's a whole cascade of variety. And so where I don't know how it's going to go, but I can see that very discussion of obviously the discussion of gendered identity and what we do to intervene in terms of a variety of different societal things. That's going to come to the forefront very soon, I think, and I think it'll be good for a wider percentage of the population to understand a little bit more than you get in the high school biology textbooks. So that's kind of the one thing. The other thing that it stands out, and there's different illustrations of it, and I think he doesn't mean to do this, but the story kind of writes itself, where he starts out talking about penetrance and what does disease mean, but then he also goes into show, for certain diseases, if we know that there's metabolic loss, you can take a supplement and basically you can have normal metabolism even if you have a certain broken pathway or something now, which really hits home at the phenotype is the product of the environment and the genotype. And I'm blind without my glasses, and 200 years ago, I would be a very deleterious allele. But now that because of easy things, society has changed to the point where that we don't think of, it stinks that we have to, wear glasses sometimes, but I'm not dying because of it right now. And I can see that for other situations happening as we go forward too, and in a variety of other ways. But it really is we can be very, say this the right way. There are, to kind of echo what others have said already, there are a lot of environmental changes we can make to have phenotypic outcomes that kind of put people on the right playing field. And it's worth kind of having those larger discussions, I think, in a bunch of different ways.
So in terms of, because I know Laura wanted to talk about this, because we're talking about interaction between genetics and environment. So Siddhartha Mukherjee tried to talk about epigenetics, and it turned, like Laura was saying, turned out confusing for her. It also turned out confusing for me. So I don't know if anyone here has more specialized knowledge in epigenetics that could explain what he was trying to do. Yes, no, I don't.
So I'll just say that I think that epigenetics to me is really important. The reason I asked Karen to especially discuss this is because it's a really interesting concept. It's a new concept. It's one that I don't have a good handle on right now, but I need to in order to be able to teach this well. But I thought, good, we're going to get to epigenetics in this book. But I had heard that he'd written an essay before the publication of this book that was not well received by people in the field. What I understand is that when the book came out, people felt like, okay, this is a little more balanced. But to me, the content on epigenetics is just really murky in this book. And we kind of get into flowery language about, isn't it amazing that we can have these things? And I'm like, no, you got to give me like concrete examples, man. We got to get into like, because I can't with the, ooh, we don't know. Well, and it might be because we just don't have a good handle on it yet. But I left this chapter, the couple chapters on it, and I was like, nope, still murky. Okay, we're going to have to work on that somewhere else.
Yeah, and I think it's murky because it's still murky, and it again comes down to this vocabulary where the term epigenetics was kind of defined in the absence of other knowledge, and then once it's very easy it's very easy to layer on new knowledge and try to make it fit into boxes that we've defined in the past, and I think epigenetics, and especially things like Lamarck and that kind of stuff, gets shoehorned in where it's not necessarily the most relevant. I mean, every new epigenetics paper in one of the glossy journals is basically like, oh, look, Lamarck was right. Well, you know, there's a large, there are a lot of heated discussions that go on, and they go on in terms of like, what is the definition of this word or that word, or how far do you go back to, because every epigenetic mark has a genetic component to it if you look at the proteins that are putting them down. And it's these philosophical traps of we're all arguing around something that none of us really understands. And it does come out in murkiness. And I think there's, we try as scientists and as humans, I think we like to fit things into nice, clean boxes, and biology doesn't obey by that. But that doesn't mean that we don't kind of shoehorn things into the boxes even when they don't quite fit perfectly. And I think that's kind of where the whole epigenetics discussion has gone to, because it is, there's definitely something that matters. Does it matter always? No. Does it matter never? No. And then we argue about, well, where do we care about it? And I think we're still at the point of trying to figure out where and why we care about it. Yeah,
So far, I guess, like, we're still sitting at that intersection of the figure that he had, which is genes, environment, chance, and triggers, or things that will, I can't remember what was in the actual figure right now, but all those things will influence how these diseases manifest, or whether or not they manifest or not. And what I found that he did stress that was useful in the book, which is that even if you look at somebody's specific genotype, that's not necessarily definitive of how they will turn out. So there's a portion and section in the work where he talks about the potential of a fate map closer to the end, where you've determined someone's genetics, and then based on their genetics, you've determined what type of person they will be, what type of personality they'll have, preferences, and et cetera, et cetera. And I never thought about it that way. Like, when I think about fate maps, I think about C. elegans fate maps, but now that you can have like a human fate map based on someone's genetics, like, that's it. What he talks about, and I think he does a really good job about this, which is that that won't necessarily happen. Like, you'll be able to have certain aspects in which you can predict, but not all of those aspects will be predictable. And to eliminate someone, or as he also spent a good time in the book talking about eugenics. So to eliminate someone because based on their genotype, is it worthy to eliminate that person because, like, of the things that they could potentially bring to society? He talked about someone who suffered from a genetic disease. I can't remember her name started with an E, and she was a presenter at a conference.
It's Erica.
Yeah. So then
she was like a teenager, late teens.
Yeah, and she had this I can't remember. She had a mutation in 2 genes, but was it a metabolic deficiency or not sure? But she's writing books. She's talking to people at conferences about her story. And to not have these people in the world, I think, would be a terrible shame. I mean, I understand that there's that conflict of, do you, what was it, the term that they used? Sorry. It was suffering. What does everyone remember? What was the other term? It was alleviating suffering, but he had a specific terminology for it. But yeah, he was right. Okay, he was doing that. And sorry, I lost my train of thought. But before I get too far, we still have probably like 1 or 2 minutes in terms of time to talk about gene therapy. So I know that this was an exciting topic for anyone. Kat, did you want to say anything about that? I remember you brought it up earlier.
Yeah, I just thought it was interesting that, well, actually this topic that you just covered brings back a recurring theme in the book, which is this: what is normal, and is the ability to predict what your phenotype is going to be from your genes really doable, and is it something that we should be trying to do, because you know, you can end up with this homogeneous population and thinking like Gattaca, or you could end up with overcorrecting. You could end up you know, not realizing the potential of people who may have a genetic predisposition for something, but it turns out that they can actually have a really fulfilling life that contributes to society. And what does that even mean? One of the things I liked about this book is that it keeps bringing back this philosophical view to this really strictly scientific idea that we have of going back to what Dave said, a gene indicates a phenotype, and explaining to people what the complications in that for genetic therapy, like it's intriguing to think that we could change, like he was talking about cystic fibrosis patients, you know, targeting a specific cell type in a particular organ for gene therapy to be able to cure this debilitating disease is intriguing, and I think it's something that we should be striving for. But I am pleased with the fact that he comes back again and again to say, okay, it's great. We have these really great things we can do. Now let's remember to keep this in the broader context of how this can be manipulated by people who have a very different idea of what they think population should look like, or where we should be striving to be as a human race. So I think it's important to keep that context in mind, and I'm glad that he keeps bringing it back up.
All right, we're just about at the end of our time. Does anyone have any closing remarks they'd like to add before? All right, so
no, me, I do.
Go ahead,
Always me. And I think we've got a little bit of time. If people really do want to have some thoughts that we haven't covered, please feel free. I mean, I don't mind staying a little bit for people to have their thoughts on this. But I just did want to say that I think, and it was brought up here at the end, is that he's got this through line where he talks about eugenics. And I think that's important, because I think this book is coming out. Finishing this book, I came away with two things. One is that it is very exciting to be at a time right now when we are learning about what is in our genes, what is in our DNA that leads to who we are as human beings. And it's very clear that this author has a tremendous amount of respect for the diversity and variety of human life. And I think that's really amazing. And the idea that we're starting to piece together what makes a person out of their DNA is something that comes through, the excitement of that comes through with the book. And so I think he's also done a really good job, as many people have said, issuing a lot of caution about, as we start to learn, then there's always an extension with people to want to control. And so then the issue becomes, think very, very carefully about how you are beginning to impose that control. And the fact that he did such a careful job going through the history of the eugenics movement, he can call back to that every time to say, you know, we always have to be thinking about the ethical considerations. We always have to be thinking about, you know, all the emphasis that he put on society in terms of how society views different combinations of genes and the phenotypes that arise from them. I thought that was really well handled. And so I think, you know, I would gladly give this book to a student who is interested in genetics, because I think they would come away with a very comprehensive understanding of the promise and the limitations and the responsibilities involved with having this better understanding of the human genome. So I didn't even write that down. There you go. Just off the top of my head.
There's it's not I don't know if it's a theme, but there's something that just, it hit me, and it comes up in a variety of different places, and then in interactions I've had with people in science. The role for moratoriums and regulations, and curse them as we may sometimes, like, you know, at some point there's thousands, if not millions, of people that are researchers out there that now have the capability to manipulate anything. And so there's this weird conflict. I'm almost finding myself rooting for CRISPR stuff not to work now, just because, like, if it's easy and, you know, you can set up regulations, but as the chapter on cloning and insulin,
there's always loopholes for the regulations, and there's ways to get around it, and there's dark money, and there's people that can do this, and or states that can do this, and other facilities kind of out of the view of the public. So I kind of almost, you know, there's this bittersweet, like I love kind of bathing in how science works and how we can do cool new things with genetics, but then there's going to be people that abuse it, and no matter what you put in, there's going to be people that abuse it, and I don't know how to deal with that, aside from I don't know how to deal with that, because it's going to happen. So yeah, there's always ways around, and there's always actors that don't have the best interests at heart of things. So I don't know, it's awesome and terrifying sometimes when I think about it, but again, I think it's great to be able to do cool new genetics things, but I think it's easy to say, you know, we need to put a governor on things and actually put regulations in place, but then I'm also not the one kind of therefore harming the careers of other people that might be setting up labs to develop such things. So it's this weird conflict that's in my head right now, at least my own head right now. But
well, on that note of CRISPR not working, there was a paper that came out probably like a few weeks ago about how doing certain CRISPR in targeted regions creates deleterious mutations in others. There was a letter in Nature about that. I'm not sure if you encountered it. I went to a conference and someone showed it to me. So, or someone.
Yeah, there's been a whole discussion after, so that depending on who you ask, that paper is completely wrong. But yeah, there's all that there's a blanking on the guy's name, Bergiano. He's on Twitter. He's got a avatar that's like a drawing. But yeah, there's a whole fervent discussion right now about the validity of those results. But yeah, I don't know which way the data falls down on yet. But yeah, I don't know. As a microbiologist, I know that it's easy to make targeted mutations, but then you always end up with other things happening at other places, and you have to kind of take into account that. So I think a lot of eukaryotic biologists and geneticists are going to discover the beauty of secondary site mutations controlling phenotypes in the next decade or so. But
all right. Well, I also really like that, Dave, that you said that it's awesome and terrifying at the same time. And I think that's a great way of ending our discussion of the book, which is that he's covered all this awesome discoveries that we found throughout understanding genes and the way that they work. But then at the same time, it's a cautionary tale, because it's also terrifying that now that we understand this, we have this power, and that we should, as in Spider-Man, harness that power responsibly. All right, and so I'll just come to the end of our book club, and so thank you so much, everyone, for coming. I really appreciate that everyone's here. And for anyone that is watching, you are welcome to join us for the next book. Laura, what are we reading next?
Next month, we're going to be reading Fatal Invention by Dorothy Roberts. So some of the issues that we talked about here about how it's kind of more the societal impact and how science and genetics are leading to issues with race, and kind of the discussion of societal views and then also the science. I've read this book before. It's very, very good. So I'm happy to read it again. So we'll be discussing that sometime in July, date, time to be determined. I try to kind of fit it around people's schedules. So I'll be kind of tweeting to get some people's feedback on when might be a good time.
All right, sounds good. So if you want to join us next time, just tweet Laura and I, and we'll send you the link information, or you'll always find the information on our website. So thanks for joining us, and we'll see you next time. Bye.